Okay, good morning to one and all present here. It's 10 o'clock now, so I hope everyone is present now. So let's start Bioseptem 2020, a Global Biotech Week celebration organized by Department of Biosciences, UC College, Alwa. So let's start. So over to you, Anjali.
Thank you, Ashama. Science has discovered that, like any other work of literature, the human genome is a text in need of commentary. For what Eliot said of poetry is also true of DNA. All meanings depend on the key of interpretation. What makes us human and what makes each of us his or her own human is not simply the genes that we have buried into our base pair, but how our cells, in dialogue with our environment, feed back to our DNA, changing the way we read ourselves. Life is a dialectic. A very good morning to one and all present today. Respected Principal Dr. David Saj Mathew, sir, Manager Reverend Father Thomas John, Speaker of the Day Dr. Surekha Zingde, other faculty members and dear participants, I invite you all wholeheartedly to Bioseptum 2020, a global biotech week organized by the Department of Biosciences, Union Christian College, Alwa. Today, we have the inauguration of the program, followed by the first talk on the topic, Proteomics to Integromics, by Dr. Surekha Zingde. She is the trustee of Indian Women Scientists Association and former Deputy Director of Advanced Center for Treatment, Research and Education in Cancer, Navi, Mumbai. Every auspicious event begins by seeking the blessings of God. I invite Gopika K. Menon for the prayer.
പ്രസാദം തരേണേ പകലിൻ്റെ നാഥ പകരം തരാമെൻ്റെ ഹൃദയം പ്രസാദം തരേണേ പകലിൻ്റെ നാഥ പകരം തരാമെൻ്റെ ഹൃദയം പുഷ്പാർജനക്കിൻ്റെ ഇഷ്ടങ്ങൾ നൽകാം പുഷ്പാർജനക്കിൻ്റെ ഇഷ്ടങ്ങൾ നൽകാം പകരം തരേണേ നിൻ കൃപകൾ പകരം തരേണേ നിൻ കൃപകൾ പ്രസാദം തരേണേ പകലിൻ്റെ നാഥ പകരം തരാമെൻ്റെ ഹൃദയം പകരം തരാമെൻ്റെ ഹൃദയം.
Thank you, Gopika. Next, I invite Mr. Shyam Mohan, Head of the Biosciences Department, UC College, for the welcome address.
Good morning to one and all. Today, I'm here to welcome all of the participants and all of the members of the community, of the biotech science community, to this program that is Bioseptem 2020. So, and of course, I would like to also formally welcome each and every one of you to this function. But before coming into that, I would first like to acknowledge the initiative taken up by the students of the biotech department, the bioscience department, for commemorating the Global Biotech Week. Never before in the history of modern world have we relied so much on the science and the awareness of science that has been communicated with the common people has never before seen such a high. So at this juncture of this pandemic, we decided to conduct this program on the basis of social distancing and in the online mode. So this Global Biotech Week, I would like to say a few words about this Global Biotech Week, where pharma companies and biotech startup companies and other entrepreneurs are involved in the awareness of science and enhancing its outreach to the world. So like I said, in this particular pandemic time, where we are so much into faith and science simultaneously, it was our goal to conduct a program to bring, not only it inspires the students to be more aware of the recent developments in biosciences, especially biotechnology, and also at the same time, the students also will get an opportunity to be active because they have been on the sidelines for some point of time. So we decided, the students also from their part had asked us, so how can we be more active? Like I remember one incident telling, how do we go, one of our students also asked, how do we survive these times of pandemic? So one best answer that I can tell is that after 10 years, maybe your children will be learning in history about how 2020 was or how the COVID situation was. So at that point of time, you should be able to tell to your child that, see, this is what I had done during that time. This is what I had gone through. So being a student and an aspirant of biotechnology, how have I contributed in the smallest way to enhancing my situation at this point of time? So from that goal, from that answer, we started our deliberations on conducting programs that would initiate the students. So it is how this particular idea was born, and it was definitely initiated by the students. So this is how we took up a program, a one-week program where they'll be actively involved, where they will listen to speakers like Dr. Surekha ma'am on advances in biotechnology from the scientific perspective. Simultaneously, they will also engage in programs like article writing, photography, quiz, etc., which will motivate them and which will give them the energy to rush through this time. So that is how this Global Biotech Week and Bioseptem 2020 was incepted. I would like to welcome now formally our principal, Dr. David Saj Mathew. Sir has been inspiration and he has been the guiding light personally for me as well as for the department. And it is with his motivation and with his blessings, we start off the program. So first I would like to welcome you, Dr. David Saj Mathew, sir, the principal of the college, to this function. Secondly, Secondly, I would like to also welcome our manager, Achan, Reverend Thomas John, sir. I believe he's a part of this meeting. Being a technical meeting, he has given us his prayers and blessings so that this program is conducted in a smooth way. And I welcome you, sir, to this function. I would like to now welcome Dr. Surekha ma'am. Surekha ma'am has been associated as was the deputy director of ACTREC, and she will be speaker for the day. She will deliver the keynote address on proteomics to integromics, which is the omics era. Most of our students, undergraduate students, will have the opportunity to understand the basics of proteomics and integromics. And Madam is a good resource person to have for us in this program. So Dr. Surekha ma'am, with a lot of respect and love, I welcome you to this program. Now I would like to welcome Dr. Susan Eapen, the adjunct professor of our department. She has been very inspirational and she has motivated us she is always there for us and wishing that we get the best resource persons, the best people to deliver important topics in our department. And her association with us has been very instrumental in organizing many programs over the years. And Susan ma'am, you continue to be the real source of inspiration, and hope our program does justice to what kind of motivation you deliver us with. Dr. Susan Eapen, I welcome you to this program. Next, I would like to welcome the teachers of my own department, Bioscience department, and the non-teaching staff of our department, as well as of the college, who has helped us in delivering this program. And I would especially like to welcome the coordinators of the Bioseptum 2020 program. They have been working in the last week tirelessly so that they accommodate all the events in the scheduled time, and all of us are not so much familiar with the online mode of programs, and they have taken up the initiative, and hopefully with God's blessings we can do it well. So I would like to welcome the teachers and coordinators of the bioscience department to this program. I also would like to welcome the outside fraternity who has been very responsive in cooperating with our program. That means the college, other departments from our college, as well as departments from outside colleges, the scientific community, the students from the other universities and colleges who are willingly happy to be a part of this program and who have lent their time to visit Dr. Surekha's talk, as well as participate in the programs that are being conducted by the department. So I welcome the fraternity outside our department, the staff and non-teaching staff of the college, of other departments, of other colleges, universities, the research scholars and students. Welcome you, all of you, to this program. Finally, I would like to say the stars of the program are the students of our department who have been relentlessly, effortlessly coordinating the online programs that are being conducted going on this week. Wishing them all of the best, because they have initiated this program. They have been the soul of this program, or the students of the BSc Biological Techniques and Specimen Preparation course, the students of the Biotechnology MSc course, the students of the Bioinformatics MSc course. It is without the help of the students, this program wouldn't have been possible. So students have it has indeed been a great effort from the students. So I'm not specially mentioning the students who are deeply associated, but I would like to welcome all of you, all the students, to this program. And once again, I wish this program is a huge success. Welcome one and all. Thank you.
Thank you for that wonderful welcome, sir. I now invite Dr. David Saj Mathew, sir, Principal UC College, for the inaugural address. Over to you, sir.
Thank you. Respected manager of the college, Reverend Thomas John Mitchell, head of the Department of Bioscience, Shyam Mohan. Today's a special invited guest, Dr. Surekha Zingde, other faculty members, non-teaching staff, and all the participants of this seminar. Very good morning to all. Okay, this is a wonderful organization, or I would like to thank this each and every person associated with this seminar. It is going to be a 1-week program in association with this Global Biotechnology Week. So the initiative is very good, and it is in the right time. The purpose of the Global Biotechnology Week is to raise the awareness of industry, this biotech industry, and its global potential, particularly to bring this awareness into the policymakers, as well as those who are influencing the policymakers, and to the general public. One way we are blessed with these technologies, particularly these proteomics, genomics, and other omic technologies. I think I feel that at the present condition, present situation, this in the pandemic situation, this much number of candidates for COVID, the vaccine candidates for the against COVID-19 came only because of the advancement we achieved in this field of biotechnology, as well as in the bioinformatics. Otherwise, we would have been in a position to in search of this drug in a very dark condition. But at present, we have many promising candidates of vaccine. That's all because, and that to come in a very short time, short period. That is all because of this technology. As we know that we started this bioinformatics in 2005. People were not much aware about this bioscience, this branch of science, and people were very much not aware at all. So I was the coordinator in those times. So really struggled to make the people understand what is actually this. But we were very sure that this subject is going to be very, very useful for the community, as well as this field is going to be flourish in the coming years. So that happened. And it is going to happen. So that is the condition of biological science. It is the era is of biology. Particularly now, all the subjects are interdisciplinary. No subject is now sticking to its own arena or sticking to its own area, but taking advantages from other areas of subjects, advantages of other subjects also. So biological science is also taking other subjects in cooperation with them and getting a very good result out of that. So organizing this biotechnology week is the right thing that bioscience department is doing. So I wish to congratulate each and every person work behind this to make this success, or going to be a success one. Especially the students, as well as your head of the department said, students work for a very, very, very hard. They work very hard to make this as this 1 week program coming to live. So that is a good gesture from the students, and particularly in this pandemic time. Also, I wish to give thanks to Dr. Susan Epen, who became the instrumental for bringing eminent personalities for speaking in this Biospectrum week, Biospectrum 2020 program. So I'm not making much. I'm not taking much time because you are all waiting to hear Surekha ma'am for the subject. So I wish to inaugurate with all your permission. I wish to inaugurate this program, 1 week program, and wishing all the best for this program, and hope that this program is going to be a very useful one to each and every participant. Once again, thank you for inviting me and giving me the opportunity to speak few words to you. Okay, once again, thanking all and wishing all the best.
Thank you so much, sir. Next on schedule, we have an introduction of our chief guest of the day by Dr. Susan Epen, adjunct faculty, UC College.
Can I have the slide projected?
Yes, ma'am. Okay. Yes.
Okay. Good morning. I have great pleasure to introduce my friend, Dr. Surekha Zingde, currently Trustee, Indian Women Scientists Association. Former Deputy Director of Advanced Center for Treatment, Research and Education in Cancer and R&D Wing of Tata Memorial Center, Mumbai. Her expertise and interest are in the fields of cancer biology, chronic myeloid leukemia, membrane proteins, signal transduction, etc. Her main research focus is on oral cancer proteomics. Dr. Zingde was also the former professor of Homi Bhabha National Institute, Mumbai. She was also chairperson of several committees like Indian Scientific Advisory Committee of Lady Tata Trust and Ethics Committee of different institutions and others. She was also a member of Research and Recognition Committee of ICT Mumbai, University of Mumbai, and a number of several government project review funding and advisory committees. Regarding her educational qualifications, she did her BSc Chemistry from Bangalore University, MSc Chemistry from IIT Bombay, and PhD in Chemistry from Boston University, USA. Later, she was a visiting fellow at Tata Institute of Fundamental Research, Mumbai. She has guided 11 PhD and 4 MSc students. Dr. Zingde has 76 peer-reviewed publications, has several book chapters, a monograph, and 4 patents to her credit. Today, we all are privileged to have Dr. Surekha Zingde with us, who will speak on proteomics to integromics. Dr. Zingde, please.
Thank you so much for that wonderful introduction, Susan ma'am. So without further ado, let's begin today's session. I invite our chief guest, Dr. Surekha Zingde, for the keynote address and the webinar. Participants, please note that you may post your doubts and queries in the chat box provided. So over to you, ma'am.
Good morning, friends. Dr. David Matthews. Can you hear me?
Yes, ma'am, you're audible. Good morning, ma'am.
Good morning. Dr. David Matthews, Reverend Matthew John, Dr. Shyam Mohan, Dr. Susan Ethan, Dr. Asha MP, faculty and students of Union Christian College, Aloha. And I note there are other students from different colleges also. Thank you for this opportunity to join the faculty and students of your college to celebrate the Biotechnology Week. As rightly pointed out by Dr. Matthews, biotechnology is the science which involves development of technologies which harness biological sources for the benefit of society. Biotechnology, as it was known then, has been going on for decades, and I really should say centuries, but it came into fore when fermentation techniques and were noted and received tremendous attention with the advent of recombinant DNA technologies. This century could well therefore be called the omics era due to the availability of the human genome sequence, efforts to decipher proteins from protein coding genes, technologies for metabolomics, and related technologies dealing with analysis of a large number of biological parameters, and obviously big data analysis. I note you have a number of programs scheduled for the whole week, and my friend and colleague of several decades, Dr. Rita Mullerkar, will be giving the lecture on the 4th of October. I congratulate the organizers of the program. I'm sure the participants will get a bird's eye view of some of the technologies being used to understand human microbes, plants, and animals at the molecular level, so that we can address the requirements for the benefit of the world at large. The best outcome of this week will be if each one of you here today, in particular the students, are motivated and get innovative ideas on how they can participate and contribute to the needs of the nation using biotechnology. In the next 30, 35, 40 minutes, I will go over my lecture on proteomics to integromics. The idea is to give you a bird's eye view of what is happening. I'm sure there is no time for in-depth technology information there, but do note down the highlights and I'm sure you will get the details therefrom. I will just upload my presentation now. Who's Anjali is helping me, or Ashwin?
Ma'am, if you are uploading from that side, no issue.
I'll load it from here, please. Okay. It's easier for me to then coordinate. Shall I do that? You have made me a co-host?
Yeah, you can now do a screen share.
Okay. Not yet. I'm still disabled.
Okay, try now.
Yeah. Can you see that?
Yes, ma'am. Yes.
Is that clear, or should I make it full screen and then go?
Ma'am,
Let's make it full screen.
Okay, I'll just make it full screen first and then I'll show it. I think that will be better. I think when I go full screen, it doesn't show me how to load. Can you see that? Not yet? Hello?
You can
You can see it? Yeah, okay.
But not in the presentation mode.
When I put it in the presentation mode, I could not load it up.
Go to the slideshow option above and start from the beginning.
Go to the
Slideshow option above in the near the animations and things. You can see a slideshow option.
Sorry.
Ma'am, at the top of the window, slideshow.
Here. And annotate remote control. More.
No, ma'am. In the PowerPoint window, you can see animation, slideshow, review, view, acrobat, like in that slideshow.
This one, no? Yeah. Okay. Yeah.
Yeah.
All right. So sorry for that little disruption. I think in this digitized world, we are learning how to handle many of the things. So I'll begin my lecture. As mentioned, this topic will cover and take you through from not only proteomics to integromics, but from proteins to proteomics to integromics. And I need to rush you through some little systems of biology to show you how biology has really changed. We were learning about systems biology when we learned anatomy, the lymphatic system, the respiratory system, etc. And we were not satisfied. Then we moved on to cell biology because we wanted to know what are the cells that make up the anatomy. From the cell biology, we moved on to molecular and cell biology because we started looking at protein molecules and we wanted to know how they actually control the functioning of the cell. So it became molecular cell biology. And then came the era of reductionist biology when we wanted to know the sequences of the gene, the sequence of the protein, to really understand what these sequences mean and how they control the functioning of our body. So from reductionist biology, which took place for almost a decade, we then moved on to the omics technologies, which was in the early 2000s, and which included not only genomics, but also proteins and proteomics, wherein we wanted to learn about protein and protein interactions. We wanted to know what are the network analysis, how a signal given by, say, a growth factor would stimulate the genes downstream. And then finally, today we are at what we refer to as molecular systems biology. So we moved from anatomical systems biology to molecular systems biology. And we still are interested in being able to run and do well. And we want to know what are the molecules that make up our cells, how these cells make up the whole system and make us function well. So this is the direction in which the science has moved over the years. Just to take you back a little bit, in terms of the central dogma, you will see here that the genes have exons and introns. The compilation of all the genes in our body is the science of genomics. And alterations in these genes give us markers which are essential to identify any clinical conditions. The genes are transcribed to make mRNA, and all the messenger RNAs compiled together is the era of is the science of transcriptomics. The messenger RNAs are then translated into proteins, and how many messenger RNAs are there, how many proteins are made, is the questions we will ask in the next few slides. And the realm of knowing all the proteins together is proteomics. These proteins, many of them are enzymes. They provide the metabolites that are required to make our bodies function. And that's the realm of metabolomics. And we are back now to looking at morphological features. We are not happy with just knowing the molecular components. We want to see what happens when any of these get altered. What is the alteration in the morphological features, which may help us understand the situation at large. The Human Genome Project was completed in 2003. That was when genomics came into play. Large number of sequences, 3 into 10 to the power 9 base pairs are available. These are in databases. The alterations in these sequences, etc., in the various genes are also recorded. So the databases are a one-stop shop to get the information that you need. Today, the question we ask is, how many of these sequences actually are protein coding genes? And the estimate is that it's around 20,000. Are they making then only 20,000 proteins to make us function so well? These are the questions we ask in proteomics. And this is a very, very challenging question, which has been taken up by the Human Proteome Project. And I will tell you more about this as we go along. So just to get back, here you can see the genome and genes around 20,000. Due to alternative transcription, you get larger number of transcripts, maybe about a lack of transcripts. These in turn make proteins due to different post-translational modifications and so forth. It's estimated there might be about 6 million proteins. We are still not there at the right number. And why do these occur? Because each of these proteins which are generated, get post-translationally modified with small chemical groups such as phosphorus, sugars, acetyl group, methyl group, and others, more than 200 or so, which make the proteins function. They may fold themselves up into monomers or dimers, or they may come together with other proteins with different functions. The folding and functional acquisition then helps them work within the cell. They could either be cytosolic proteins, they could be nuclear proteins, they could be membrane proteins, and in their relative positions, they contribute to the functions of the cell. Here I show you what are the number of the known post-translational modifications which are known today. Majority of them are phosphoproteins. Some of them are glycoproteins. Ubiquitination is another post-translational modification that is added. Acetylation and methylation. Phosphorylation occurs either on tyrosine, serine or threonine. Glycosylation occurs on asparagine or serine or threonine. Ubiquitation on lysine. Acetylation could occur on lysine, alanine, methionine, or serine, and so forth on methylation and so forth. And each protein could either have it on just one of the amino acids, it could have multiple amino acids, and the position of the post-translation modification could be very dynamic. It could change depending on the cues that the cell receives. So up till now, you would have realized that we have information about the sequences of the genes. We have emerging information about the amino acid sequence of the proteins. We are gathering information about alterations and modifications. We are gathering information of expression patterns of the messenger RNA. We have three-dimensional structures of the proteins. We have an atlas of proteins, which I will show you a little later. All these have databases. These databases are the basic requirement for the knowledge base that we have today, and which is now referred to as bioinformatics. And I understand your college also has a course on bioinformatics, and I'm hoping that this lecture will give you some idea as to how what you study in the classes will become useful for you to understand biology at large. We will now move on to the proteins per se. What information do we need to know about the proteins to understand the protein function? We need to know its biological function. We know its physical characteristics. We need to know its chemical characteristics. In physical, we need to know, of course, the primary sequence, the secondary structure, and tertiary structure. In chemical, we need to know its charge distribution, eventually leading to activity, surface hydrosolubility. And now in terms of biological, we need to know how many isoforms there are. I'm sure many of you have learned in your biology courses the term isoforms. We need to know where are these isoforms localized within the cell. We need to know how the proteins interact with each other, the interactions. We need to know where are they distributed, in which tissue are they located. Are there any post-translational modifications? How do they work in a cell cycle? A cell cycle is just given as one of the functions, but in any function, you'd like to know which protein is involved. You would like to know the turnover of the protein, that it is half-life, and you would like to know its abundance. Is the abundance static or is it dynamic? Are questions we'd like to know. So the term isoform is now actually changing. We know that the human gene can code because of the presence of exons and introns, and which of the exons come together, you may get more than one messenger RNA arising from a particular gene. Each of these messenger RNAs would be translated to form what are referred to as isoforms of the protein. But really speaking, these isoforms may either have already a single nucleotide polymorphism right in the beginning. They will give you a slight change in the protein, or these proteins may get post-translationally modified. Just two are shown here, glycosylation and phosphorylation. And each of these changes brings about different proteoforms. So instead of isoforms today, we need to be talking about how many proteoforms are formed from a single human gene. Is it just one proteoform, or are there multiple proteoforms? Are they steady and static number of proteoforms, or are they dynamic? Are some of the very challenging questions that a protein chemist has to address these days. So the proteoform then gets together with other proteins, forms a protein complex. These together form an organelle. The organelles then become part of a cell. The cell becomes part of a tissue. The tissue then is part of an organ and the organ system at large. So the connection between proteoforms to organ system is very clear here. Very simplistically shown, we don't know how many proteins are there in these protein complexes. We do not know how, we are aware morphologically how many organelles are there, but what are the proteins in each of these organelles are again questions that are being addressed. So the challenges that a protein scientist faces today are: what are the number of isoforms from a single human protein coding gene? What are the number of proteoforms? What are the structures of these proteins? What is the location? What is their expression? What is the amount? What is their turnover? The dynamics and the interactions of these proteins, and many other parameters which eventually lead to give the functions of a cell and of the whole human being. So within the lifetime of a cell, the protein components change their location. They may change in abundance. They undergo modifications. They interact with others. And these cannot be followed by tracking messenger RNAs alone. We need proteomic tools to understand the status of how the single proteins interact with other proteins. So we need to know all the proteins and therefore the proteome. We need to understand the static condition, and then what are the alterations that take place in the dynamic conditions. Many other such questions. And this is where the Human Proteome Project comes in. It was, the HUPO was started way back in 2001, and the Human Proteome Project per se was started in 2010. And there are three major pillars. They call them the three working proteomic pillars, which have made it possible for us to ask the challenging questions that I addressed before. These are the mass spectrometry, the technologies related to mass spectrometry, technologies raised to antibody development, technology to knowledge base of bioinformatics. And each of these technologies have moved forward very rapidly. And that was the case way back in 2011. And it was anticipated that by 2015, we would have some information. Now we have 2018, wherein we not only need these three pillars, we need to know the pathology or the histology of the tissue to be able to opine more information about the protein and its function. So to go back, HUPO is the Human Proteome Project. These are the three pillars that I have been talking about. These three pillars are the main workhorses for the Biology and Disease Human Protein Project and the Chromosome Human Protein Project. And these are helped by the different bioinformatic support systems, which comes from UniProt, Protein Exchange, Peptide Atlas, and the Human Protein Atlas, which is actually not a bioinformatics source, but another atlas, which I will show you now. So biology and disease, chromosome, they work together to tell us more about the proteins. The biology and disease driven human proteome project, actually there are 19 groups which are looking at different aspects. They're looking at cancer, cardiovascular, diabetes, rheumatic disorders, stem cells. They're looking at the mitochondria, musculoskeletal, and these are across the globe. And they are coordinated through the Human Proteome Organization. Then we have the chromosome-centric Human Proteome Project, and these are 25 groups. Many countries are involved again here. The goals are again to obtain the identity, abundance, subcellular localization, same as that for the biology and disease. There they are with respect to a particular disease or biology system, and we need to identify at least one representative protein among the 20,000 predicted protein coding genes in a chromosome by chromosome manner. And here India is taking the lead with Dr. Ravi Deshmukh, who's presently was at the CCMB before, now is with the Institute of Bioinformatics and the Mazumdar Shaw Center in Bangalore. So he leads a group for the work on chromosome 12, and he has other members of the Proteomic Society India from different institutions who form part of this team, along with other global members too. Then I come to the knowledge resource pillar of the HPP. And this is the Human Proteome Standards Initiative, which means that anybody doing proteomic work gets mass spec data or any other protein related data, has to put it into what is known as the Proteome Exchange and another forum, GPMD. And from here, the data is reanalyzed and the raw data and metadata is checked out and curated and made available in the next brought form and could be used for publications or for guidelines. So today you cannot just collect data just any way you want. There are definite mass spec guidelines as to how you have to collect the data, how you have to annotate them, and then submit them to the for peer review. So from the Human Protein projects, we have now reached and got 17,874 proteins. And the total predicted at this point, January 2020, you will find on HUPO website that we expect there are 19,773 single protein coding genes, and we expect at least this minimal number of proteins. Many of the proteins are present in small amount, not detectable by mass spec, and they are low transcripts. They may not have lysines and arginines, which are required for mass spec analysis. And each of these are categorized based on their level of evidence, protein level of evidence. One is when they have good mass spec data telling that this protein does exist, and some other parameters. And these are this number of proteins. There are others where a transcript is seen, but we don't see a protein. there is some homology and some are predicted. These are these numbers. And at this point, HUPO claims that we have the 90% of the human proteome has been discovered with respect to human protein coding genes. Has India contributed to those efforts? Yes. Our Dr. Shubhraj Chakravarti, Dr. Ravi Sardeshmukh, Sanjeev Srivastava are members of the HUPO councils. We have the Asia Oceania Human Proteome Organization, which are a body of proteomic societies in the oceanic region. And Dr. Shantanu Sengupta is presently representing us. Then there is a plant proteome organization where Dr. Renu Deswal is there. There is another Asia Ocean Agricultural Proteomic Organization where Dr. Shubhra Chakravarti is again representing. And the Protein Atlas project is represented by Sanjay Navani. I think I missed out. I missed out on this protein. I did not explain to you. This Swedish human protein project actually it's a beautiful site that you should go to. The antibodies have been raised against almost 26,371 antibodies are there against 17,000 of the proteins. So now you know that we've nearly got that many number of proteins identified. And the presence of these proteins in normal and cancer tissue is there. Their subcellular localization is known. They're also looking for messenger RNA levels and for messenger RNA isoforms. So do visit this site. And if you put in the Google Human Protein Atlas, you'll reach it. You can keep the whole week for yourself to investigate that site. Now I'll move on to proteomic approaches. And here, if you see, these are some of the techniques that you have been doing in your laboratories, and they are simplified so that you can be with me as I keep talking. If you take any tissue, whether it be a cell or it can be a tissue piece from the human body or a body fluid, could be blood, plasma, urine, and extract the proteins, these could be separated either by SDS page, which you are all familiar with. And this is separating only on basis of molecular weight. If you want to do better separation, you do separation first in the direction of their isoelectric points and their charge, and then on the molecular weight. So the same proteins resolve better. If you want to know which are these proteins in each of these bands or in each of these spots, you take them out, extract them, do a trypsin digestion, and then put them through a mass spectrometer. And how do we analyze and identify through a mass spectrometer? I will tell you shortly. Sometimes the number of proteins within the tissue or the cell or the body fluid is so many, you cannot handle the data that's emerging in the mass spec. So you need to enrich or you need to fractionate the information. So enrichment can be done. So if you're interested in looking at only some proteins which have specific affinity for a ligand, or you need to look at the protein which are phosphorylated, you can do a phospho analysis. You can bind them to a column which has affinity for phosphor groups. And these enriched peptides are then put through a mass spec for identity. Or you could take the mixture here, fractionated by column chromatography, and then the fractions could be studied further. Here is another way of looking at the number of proteins in your sample. Very often you will have condition 1, a protein, let us say, or a cell type, and then you put, let us say, a hormone in another petri dish to see what is the effect of the hormone. And you may have a third one where you put a different concentration of the hormone. So you have 3 conditions where you have unstimulated cells stimulated with, say, 1, stimulation 2. The proteins are then labeled during the stimulation period with cytofluors, which are referred to as Site 2, 3, and 5. And you mix all of these, you can run it on 1 gel. You may well ask, why did we not run it on 3 gels? Because very often happens when you try to make 3 gels, they don't come out exactly the same. and the relative differences may be different, and it would be difficult to identify the proteins, whether this protein is the same in the first condition, second condition, and the third one. When you mix them, you are running them all together on one gel, then the gel picture can be looked at by the emission emerging from the cytofluores, and for each of them you will get an image, and from the image you can make out which protein is expressed to which amount, or is it decreased or increased in amount due to the stimulant that you have added, and that will help you to understand and study the question that you have raised. And this is another method which is referred to as two-dimensional difference in gel electrophoresis. So to simplify whatever I told you up till now, you take the sample. It could be tissue or a biofluid. You can extract the proteins. You can separate the proteins through gel-based methods, or you could separate the proteins by chromatography-based methods. And you can do mass spectrometry, and from the mass spectrometry analysis, you can get information which is then put into databases, which helps you identify the proteins. The chromatography could be based on size, charge, hydrophilicity, or on affinity. This is what the mass spectrometry actually does. The digest that you have is either separated by what is referred to electron spray ionization or matrix assisted laser deionization method. So this ion source could be either of the two type for proteomics. These peptides are put into this, they are ionized to generate peptide ions. These peptide ions have a mass by charge value, which are then separated through an analyzer and then detected. And the detected information gives you the identity of the protein through a database. In matrix assisted deionization, the protein is put here. It is mixed with an chemical, which on putting a laser helps the ionization of the protein peptides, which are then put through the analyzer and then detected. And now there are not only a triple quadrupole and time of flight separate machines, there are triple quadrupole along with time of flight. There are machines which are called as orbit traps, there are Fourier transform, and very high-end machines which are being used for detection and analysis. So mass spectrometry is used for identification, quantitation, and for targeted identification of specific proteins. Just to run you through again, the cells and tissues, we get a protein mixture, we can separate them, they're digested into peptides. The peptide mixture is separated through either liquid chromatography, then they are ionized, and the ion peptides are pushed through a mass spec. And this is the kind of peak pattern that you can get, which we call as a fingerprint. This fingerprint is then put to the database, and it helps us to identify what is the protein. There are different ways of labeling this protein mixture, either with isotopes or tags, and depending on the tagging system, these fingerprints will change, and that change can be used for quantitating the changes in the amount of the protein between, say, a control condition and a treated condition. In a targeted mass spectrometry, instead of looking at all the proteins together, we can look at specific proteins by regulating the parameters on the mass spectrometer so that only the peak corresponding to that protein is seen. So mass spectrometry is very versatile instrument and the basic instrument required for proteomics. Now, having found out how many proteins are there, you would like to know how many proteins that the whole tissue have. Not every time can you be doing mass spectrometry. If you know there are, say, 100 proteins in a particular sample, and you want to look for them in other different samples, how many of these 100 proteins are present in 100 different samples, then you can do different types of microarrays. And these are done by either putting the antibody against each of the 100 proteins, layering them with the cytosolic material that you have bound, which is tagged, and measuring the fluorescence that is obtained. And similarly, if this is not clear enough, you can increase the sensitivity by using a secondary antibody also. And many of these things are done on a small little chip where over 1,000 spots can be put. So you can analyze really about 1,000 samples at a time, look to see whether a protein is present in all of them, what is the level of the protein that is present in them, is it less or is it more, and then you can use it for your analysis and understanding the biological query that you have in mind. In the reverse phase protein array, you generally put the cytosol, the total cytosol here or the fractionated cytosol, and then look for it with an antibody. So it's just a reverse process. But this gives you, in a one little plate or a chip, you can identify how many proteins are there in a multiple way. The other technique which is very useful is the tissue array. Very often you do not have tissue samples, especially in a hospital or elsewhere. You may find that tissues are embedded in paraffin. So we call this as a paraffin block, or we'll call this here as a tissue block, donor block. And this is we take portions of this with a little tube and punch out the region we want, transfer it to a recipient block. We'll do that for multiple tissues. You may have hundreds of tissues aligned like this. Each of these corresponds to one tissue. And so you have on a little chip, all these 100 tissues placed. These are then sectioned. Those of you who do histology for your microbiology experiments or experiments in the laboratory, you will put the thin sections on a slide and it will look like this when you look under the microscope. And this slide can then be stained with the stain that you're interested in or with the antibody that you're looking to see which protein is present in which particular sample, because each of these little dots is one sample. So you can analyze so many samples at one time. So that's why it's called a tissue array. The next challenge is, yes, I can look at so many samples together, but how much time will it take? So here, this is an example of how efforts are being made. And here it shows that if you want to know what are the proteins in the plasma, you can start with even 5 microliters of blood, from which you'll get just 1 microliter of plasma. You can do the protein digestion and put it to the mass spec. You'll get the raw data, analyze the raw data, and then you could do it for multiple number of individuals who probably have a particular disease condition, and then find out what is the differences in the plasma proteome in all of these. Do they have a particular odd protein which should not be there in them, and how do I treat them? So it's, at the moment, it's anticipated from collection to finding a data, getting the report, it may be something like 3 hours, and they're all working towards making this timeline even faster. So proteomic studies are addressing a large number of tissue types and diseases. Information is emerging about potential markers from each of these. We also have the protein profiles from mass spec for about 95% of the predicted proteins. These have been done by making peptides. And assays and instrumentation are being developed. As you see, what you have, I've shown you very simple ones, but the more sophisticated instrumentation to do each of those techniques which I have shown you. You noted that we have antibodies for almost 26,000 antibodies targeting to 17,000 unique proteins. How should we use this to bridge the gap between the bench and the clinic? What are the opportunities and challenges that we face? Some of them I have shown you. I will show you a little more quickly in the next 5 minutes or so. So I'm just taking a case of a protein biomarker for disease management. We really have to find out in a particular disease what are the altered proteins. We have to verify that these are definitely representative of the alteration, and we have to validate them in a larger number of people who have the same condition. So what we do is we start with about 10 or 50 individuals who have a particular disorder. Let us say they have a, tissue, oral cancer, let us say. You take the tissue out and you do shotgun proteomics. And from all the 50 individuals, you can identify about 800 proteins which are repeatedly seen. You need to verify this, that these 800 proteins generally represent the disease condition. So verification is done by using the techniques that I mentioned to you just earlier. And this verification is done on a larger number of samples, samples from different individuals. And you'll find that maybe these 100 potential markers come down to only 10. These 10 have to be further validated. And when you do validation, you'd like to use very simple immunoassays or so, which can be used in any laboratory. And those can be tested on a larger cohort of people. And even then, from these 10s, you may just come down to 2 or 3 markers which are available for the test run. So I've just put this in another way: discovery, verification, validation, and again, clinical validation. When I say clinical validation, this is within your own laboratory. This is across institutions. That means then only can you say an X protein is a representative of a disease condition. So when you do something at the bench, you must ask the right clinical question. What is the sample to be used? Should it be tissue or biofluid? How easy is it to collect? You cannot be doing surgery all the time. You need to let us say it's easy to take plasma, blood, or it's easy to collect urine. What are the technologies to be used? What is the sensitivity and specificity of the test? How easy to use in the laboratory? How much time it takes from collection to reporting, the one that I showed you earlier? How much of sample is required? What is the technical reproducibly intra and inter-laboratory? This is very, very important. These are the challenges one has to face. And in the clinics, you have to connect with the doctor to give you a proper study design. What are the patients you should be taking the sample from? What are the endpoints you should be studying? And unless you work in hand with the clinician, no question is right. And thereafter, when a marker becomes most possible, does it go for drug development and disease management. I'll take you to another place. We moved from proteins. Now we are integrating proteins to genomics. You would have heard of the Cancer Genome Atlas, where there are a large number of tissue samples from cancers, which have been genotypically annotated. Now this program, CPTAC, is of the National Cancer Institutes, took some of these samples and tried to see if the genomic alterations pan out with the protein changes. And this is what they have found. And they are sharing these resources with individuals to see how far we can go and understand the disease process well. This is the area of proteogenomics. So we're bringing two different omics together. And this program has now become international. India has also joined them. A team from the Indian Institute of Technology, Bombay, and the Tata Memorial Center have joined the NCI efforts for proteogenomics. We are looking together on oral, breast, and cervix cancers for patient care and public dissemination for information sharing. So today, whatever we've done is we've studied the steady state of the cell proteome in one single experiment. We would have done it with or without fractionation. What you've got is average data from a heterogeneous population of cells. You know very well that even in the intestine or the brain, there are different types of cells. So when you do any analysis, you take a brain tissue or a piece of the intestine, you are not really taking a cell, you're taking a heterogeneous population of cells. So the next gen proteomics proposes to look at each cell separately. And that's where the Human Cell Atlas came into being. It was proposed in 2017. It's working very fast. It's looking at decoding the genes which are active in each cell, and they're going to do omics in different cell types within different organs within the body and trace the molecular circuitry between them and try to see how they actually function together. So we've now moved from heterogeneous to single. This is how they take out a single cell under the microscope, extract the protein, separate it on a capillary electrophoresis, do a mass spec and identify the proteins which are there. This depicts it in cartoon diagram, the same thing. It's called single cell omics. So look at the way we are going. We started with systems biology, we went to reductions biology, we've come back to single cell biology. And there are many methods which I'll not go into detail here, but it's worth to remember that these are different technologies being coming into fore to study a single cell from each of the systems that you want. Now, from a single cell, we still realize the single cell is part. You still have to do omics. You still have to understand whether genomic information is converted into the transcriptome, the proteome, and then the metabolome, and so forth. So these have to be addressed to get the holistic picture of the condition within the system that you're studying. When I say system, I don't mean human alone. You could be studying C. elegans, you could be studying a plant, you could be studying Drosophila, you could be studying anything else. It's important to see what is the question that you're asking. Use such a, what is the biological question you're addressing? Choose an appropriate technique, whether you want to do sequencing for genomics, whether you want to do exome sequencing to get RNA information, or mass spectrometry for protein, or do you want to do mass spectrometry and NMR spectrum for metabolomics? And then which of these you would like to integrate to get appropriate information to understand the clinical question you have in hand. So this is really about integrating all the omics data and the appropriate omics data. The first part is the first two questions. Which is the question you are addressing? Which is the technology that you should use? And therefore, today, if we have to move ahead, I'm talking in terms of the clinics. We need to have molecular data, which would be transcriptomics, proteomics, reverse phase arrays. We have to enrich the proteins and so forth. We have to clinical phenotype, which is the survival analysis. How is the patient faring? And we have to have the pathological features, the image features. All of these together have to be integrated to ensure that we have improved patient outcome. Otherwise, just doing one of these and saying this is the right protein which will help us treat the patient may not be correct. You may have given a particular drug, you will find that it may helpful for one patient, but another patient with the same disease may not respond accordingly. And that time you have to put on your thinking caps to see what is it that we need to follow up. And that's where Integromics comes to fore. So we need to do basic research. We have to work with the clinics for clinical research to ensure that this basic research actually becomes translational and can be taken out to the public at large, out to the community. And working hand in hand is a must. The bench scientists, clinicians must interact. Then eventually with academia and industry, community workers and other physicians. Many of you will realize this is a very important phenomena. What you do in the four walls of your laboratory is insufficient. Asking the right question, addressing in with the right technologies is the only way forward to ensure that you use the technology relevant to harness the biological resource that you're using for the benefit of the society at large. So I'd like to thank you for your attention, and I hope you have been able to stay with me right through the lecture. I'm open to any other questions that you may have.
That was indeed a wonderful session, ma'am. Thank you for taking your time to throw the door of proteomics and integromics wide open for us all. I'm sure that all of our participants are waiting eagerly to clear their doubts and queries. The participants who have their questions, please type your questions in the comment section.
You want me to look at the chat box and answer them directly, or is it somebody coming forward to read them out? Okay,
Yes,
I
ma'am,
see.
I am going to ask.
You are going to read them out? Okay.
Yes.
Hello.
Ma'am, can you hear me?
Yes, yeah. Yes, I can hear you.
Ma'am, it is said that more than 90% of human genome found to be in transcript, and many are supposed to get translated at very minute levels. Is there any approaches to find them out?
See,
And the question is by Reshma James.
So you're saying that you may see them at the transcript level, but you may not see the protein, right? Can you repeat the question? Hello.
Yes, ma'am.
Yeah, please repeat the question.
It said that more than 90% of human genome found to be transcribed, and many are supposed to get translated at very minute levels. Is there any approaches to find them out?
See, that is the way we have to look at the biological question. We are missing many proteins because we haven't got the tissue or the biofluid at the time when this particular protein is available. And if it is available in very minute amounts, though the transcript may be present, it may not be made at that particular time unless it is stimulated by a particular cue, maybe by a signal coming from another cell, a growth factor signal, or an environmental signal. So that's one of the major reasons why the transcript is seen, but you may not see the protein. What you're saying is right. Yes, these are very challenging questions. I understand the olfactory proteins are very difficult to catch for this very reason. Unless you have the right smell at the right time, you probably don't see the protein which is necessary to smell it. Yeah, can we take the next question?
Okay, thank you, ma'am. The next question is by Divya Menon, and the question is, does CPTAC include data from research done in India?
Right now, when it was CPTAC, that time India was not involved in the ICPC program, which we have recently joined. What is this initiated? So I wouldn't be able to tell you directly whether there is information. So I'm sure in a year's time or so, there would be information that would emerge. Right now, there might not be. Hello.
Any other questions?
Ma'am, I would like to just ask you a question.
Yeah.
This is Shyam.
Yeah.
Ma'am, how crucial is the role of protein folding in proteome, in what we analyze, the part we are analyzing now? Has there been consistent efforts to understand the role of protein folding in proteomics?
See,
in the
let's distinguish two things. When we do proteomics, we're really taking the protein and breaking it down to identify it. And the folding component comes when the protein is actually functional. So we are looking at two different aspects of it. So if you want to know the identity of the proteins, whether it's folded, you really have to unfold it to be able to identify it, because you need to do mass spec analysis or tryptic digestion for it. But to understand its function, it should be folded in its proper orientation, interaction, so that can interact with other proteins, or it forms dimers of itself, so that it can contribute to the function that is related to it. So you have to look at it two different things. One is for function and one is for the identity. So let's understand the term proteomics. Proteomics includes all these different parameters, but what is it that you're looking for?
Thank you, ma'am. Thank you.
Good morning, ma'am. I would like to ask you a question. My name is Divya Menon, and I'm presently doing my research on thyroid cancer. So my work is mainly involved on a transporter protein, sodium iodide transporter specifically, which is used, which is basically involved in intake of iodide into the thyroid gland for the synthesis of thyroid hormones. So it has been understood that in thyroid cancer, this particular membrane protein gets expression is deregulated. It gets sequestered into the cytoplasm more than being on the membrane. So my work is understanding the role of a particular mutation, the beta V600E, which is more commonly present in thyroid cancer, and the relationship between that mutation and this protein. So actually listening to your talk, seeing the various methods that omics can play in this, I wanted a word of advice from you as to how I basically do molecular work completely. I work on cell lines. I wanted a bit of advice from you as to what would be a good method for me, because now I've started, I plan to do a bit of bioinformatics work too in my research. So listening to your talk, proteomics plays a huge role. So I am harnessing information from the TCGA also to do this, but listening to your talk about CPTAC, what would be a good approach that I should take up?
So you're, from what I understand, you're looking at a protein where in which you only have one mutation at the moment, right?
Yeah, sorry, ma'am. The protein doesn't have a mutation. The mutation.
The gene has.
Yeah. Means what I mean is it's an downstream. This protein gets, deregulated.
I got it. So the protein that is made from the genetically modified gene, it would also have the alteration. So you since you know the genome sequence, you would be able to know where the protein is altered, correct?
Yeah, ma'am, correctly.
And then you would know where is the wild type protein and the mutant protein. You will know how
yes
by bioinformatics or modeling studies.
Yeah.
And then from the modeling studies, you will be able to know whether it is functioning properly and is it folding properly to be able to do its function as a importer.
Yes.
Correct?
Yes, ma'am.
So if you have the protein with the mutant protein, you should be able to model it. That will give you an idea as to how it is malfunctioning or it's not folding appropriately. And then you could go ahead with whatever else you need to do. Does that answer your question? And
yes, ma'am.
Because I think if you already have the genetic alteration in hand, and you also know the protein which is made, what is altered in it, then you can model it and then see how it's going to function or malfunction, I should say.
Yeah, because presently the problem with this particular cancer is that as the cancer progresses, it's when this protein gets completely deregulated. Like the aggressive version of this cancer has a very deregulated version of this protein. So my basically study I want to know is how and what leads to this malfunctioning, like probably this post-translational modifications. So I can upload
you need to do a little more of bioinformatics survey to find out, is this protein interacting with other proteins for its functions? Is it having different PTMs on it? And what is the status of these PTMs? And if the answers are not there, then those are some things that you need to identify. And you
So I want to know if,
you may be having the information on the net already. If it's
that's what I was going to ask, that I should be able to get this information from either CPTAC or any of the omics programs that you just explained.
I think any of the databases probably would already have it if it's already cataloged. You don't need to go to CPTAC per se.
Okay.
If it's altered, you can be able to see. And even the protein atlas would be a good place to start to see for this particular protein, which is already well known. What is the status of it? What are the PTMs that are already known? And then you could decide what else is not known for you to pursue your PhD program. Yeah.
Okay. Thank you so much, ma'am. It was a beautiful presentation. It was very informative. Thank you.
Thank you, dear.
Hello, ma'am.
Yes.
We have a question from one of the participants. They are asking, there are many short proteins, like they play important roles in signaling and immune responses. So what would be the approaches to characterize them?
When you say short proteins, what is the length of your short?
I don't know the person who is asking the question. I think maybe they're referring to a very small peptide proteins.
See, you can catch them even in the mass spec. That's not an issue. If you're able to get, harness them and have them in solution, you can look at them by mass spectrometry and identify the protein and even quantitate them. And if it's a known protein, even there might be assays by ELISA, which would tell you how much of it is there or is it present.
Okay. We also have another question from Shraddha Mahindra. She is asking, how come proteomic profiling will help to develop predictive biomarkers for breast cancer?
See, when you do for any cancer, I won't say for breast cancer per se, because for any cancerous tissue, if you must have a normal counterpart. So if you take breast, let us say, you should have an area of the breast which is not histologically involved, or what the pathologist says is not involved. You do the proteome analysis from both of them, and you can find out, you'll do the proteome analysis of the histologically normal tissue and that of taken out from the breast. Extract the proteins, digest them, get the mass spec analysis, identify the protein components present in them, see which are differentially expressed. And then if you do this for a number of patients who have the similar cancer, today breast cancer of different subtypes. So when you're asking me a question like just breast cancer, I'm just answering you very naively, assuming that we will not look at the subtypes. So the altered proteins should be present in, let us say, 10 or 15 more other patients' tissues also. Then you can say that these are proteins which are altered. You need to understand which are these proteins, what is their function, what is the likelihood that they are regulating the alterations that bring about transformation, and then study it further.
Okay, thank you. Thanks a lot, ma'am. Yeah. So I think that was the last of the questions. I don't see another questions popping up in the chat box, so I think we'll round them up. So thank you so much, ma'am, for your patience and for taking time out to educate us on the area of proteomics and integromics. Thanks a lot. Participants, please note the link for the feedback form will be available in the chat box. Kindly fill it up within 15 minutes. Please note that the link is available only for the registered participants. Now I invite Mrs. Geetu Paul for the vote of thanks. Over to you, ma'am.
Good afternoon, one and all. Honorable Chief Guest, Dr. Surekha Zingde, Principal Dr.David Saj Mathew, Dr. Susan Epen, the Adjunct Faculty of UC College, Shamsar Avrechodi, Mrs. Asha MP, Coordinator of the program, and all others present here. It is my privilege to deliver a vote of thanks to all on this auspicious occasion. On the behalf of the Bioscience Department and the Bioseptem team, I extend my very hearty vote of thanks to our chief guest, Dr. Surekha ma'am, for gracing your important work and sharing with us your findings and opinion today. Thank you, ma'am, for throwing lights on the transition of the research interest from basic human genome to proteomics and then to the productive Integromics, where we are integrating all the omics. Thank you, ma'am, for your valuable session. Next, let me show my gratitude to Dr. David Saj Mathew, Principal UC College, for providing encouragement for organizing this event. Sir, always try to foster all these type of efforts by all the departments. Further, we are grateful to Dr. Susan Epan, the adjunct faculty of UC College. You were there all the time to guide us with your expert knowledge and experience. Yesterday, there was a mock meeting and you spared your valuable time to give suitable instruction for the proper channeling of today's program. So now, next, let me thank Shyam sir, who is a pillar of our department. He provides us with perfect logistics support and guidance. Thank you, sir. Then actually, I don't know how to render a formal vote of thanks to Mrs. Asha MP, who's a coordinator of this program. Actually, she round the clock to make this event a grand success. Each time she is thinking of doing something which could beautify the event, and she always trying to be cautious about the transparency of the program too. So thank you, ma'am. So now let me acknowledge my colleagues, the Faculty of Bioscience Department, for extending their support, for especially in judging the competition which is going to happen in the following days. A special mention to Dr. Serene Zara John is very important because she was always there to comment each and every activities from our side. So now students, I can't forget you. You're the backbone of this event. From the head to tail, you took all the responsibilities. Now I would like to give some special mentions. That is the Ashwin and Nadesh who are handling the technical section. Then Revati, Anupama, Arati, Anjali, Anita, Sumit, and all thanks for their excellent involvement in this program. So sorry if I missed any of your names. Now, last but not the least, I would like to thank all the participants. Actually, we got a huge response from the participants all over India. Even within a few hours of announcing our program, we get a huge response, and that was really a encouragement, and that provided a confidence and strength to our mind to host this program. And thank you once again. So let me wind up. Thank you all.
Thank you so much, ma'am. Participants, kindly note, fill in the feedback form as soon as possible. So that was the end of today's session. I thank each and every one of you for taking your time out to attend our session. Good day to you all. Thank you once again.
Thank you. Thank you so much.
Surekha. Thank you, Surekha.
Thank you, sir. Thank you, sir.
Thank you so much. Okay.